Hello, welcome to the November 2015 Getting Started with iPlant webinar. I'm Jason Williams, coming to you from Cold Spring Harbor Laboratory. This webinar is a monthly series that we have to invite all of the somewhere between 500 and 700 new users we see getting an account every month in iPlant, just to give you a chance to be able to know what comes with the account, know what some of the capabilities are, maybe even some that you haven't explored yet, and give you some tutorials that help get you started to using the iPlant resources and tools. So over the next hour, I'll give you a little bit of a quick background into what the iPlant project is about. Then next, I'll give you some practical hands-on tutorials that you can use to get started using the features of your account right away. Before I actually begin with those demos and with the little bit of history, I wanted to make you aware of some of the features of the Adobe Connect technology that we're using to broadcast this to you. So first of all, if you have any questions, at the bottom of the screen you're going to see that there's actually a little panel or question where you can actually send your questions to me at any time. And then myself, off camera, I can actually go through any questions that I've received and then would be able to answer them for you right away. This is one of the advantages of doing something live. As many questions as you might have, please at any time interrupt me, stop me, and let me know what I can do to help you with a particular question or something you'd like to see more of or see again. Additionally, in this room, you'll notice that on the side of the screen there are a couple of links. There are some downloads at the top. There are also some links at the bottom that'd be useful for you to explore. We'll be going through some of those, but if you want to download the Getting Started guide or a copy of the slides, you're welcome to do that at any time. Finally, really important, at the top of the screen there's actually also a little button with 4 arrows in it, and you can actually click that and make the video full screen. This is important because especially when I start to do some of the stuff on the computer, that demo or the screen may be small, and if you choose to go full screen you might be able to see it a little bit better. Also, if you are on a connection that maybe is not that fast, when you move your mouse over the video itself, you can actually adjust the quality. So if you wanted to make it a lower quality instead of HD, that would increase or that make the bandwidth used a little bit better, and you might have something that's not choppy. Okay, so hopefully you have no technical glitches. If you have any questions, as always, just let us know below. And let's get started with a little bit of an introduction to what is iPlant as a project. Okay, so I have a couple of slides for you, and the first one is actually a little bit about the vision of iPlant as a project. As a project, we have a vision because that informs all of our goals, what we're doing, what we're working with. And in this case, I've made a little bit of a cartoon. Well, one I've stolen. This is from the New Yorker. As I mentioned, I'm in New York, and this is a famous explorer standing over one of the cliffs of the Verrazano Pass over New York City, saying, "Someday this will all be infrastructure." And Verrazano, that's actually the name of the explorer, is sort of pointing. In this case, I've chosen the image of Rosalind Franklin's double helix there. And kind of the question is, who would have known 50 years ago, a little bit more than, when the structure of DNA was being deduced, the importance of it was being understood, who would have known that working with biology today doesn't mean necessarily being in the lab with tubes and centrifuges, but even more so means working with analyzing data. And in fact, I think this quote that someday this will all be infrastructure is quite true, especially in the case of iPlant, because as it turns out, in order to get people where you want to go in science today, you need cyber infrastructure. Now, in the case of Verrazano, in the case of building a city, you need that bridge, but it's actually those bridges, roads, and tunnels that build a city, which also for us in science are going to help us to bridge the gap between the biological wet lab world and the technological world of cyber infrastructure, bioinformatics, data analysis. And so in words, our vision is really to enable life science researchers and educators to use and extend that cyber infrastructure that we've built, and using it to understand and predict the complexity of biological systems, which we know are dynamic, and also are interrelated intimately with the environment. But what is cyber infrastructure? Well, cyber infrastructure, in one sense, is not all that unfamiliar. it's many things that you already probably use on a day-to-day basis. The ability to store your data, software, high-performance computing, people. These are all elements of cyber infrastructure. But our goal as a project is to organize these elements into systems that you can use as a biologist to solve problems of size and scope that would not otherwise be solvable. So in other words, as a researcher operating in today's climate of not only doing work at the bench or in the field, but now also using technological means, data analysis to understand your projects, our goal as a project is to allow you to answer questions you may not have thought you could answer because there were too many genes involved, too many systems, the data sets were too big. The acquisition of data is becoming incredibly facile. We're able to get lots more data sets than we could have before, but analyzing it, storing it, moving it, managing it, all those other hard pieces are what we think iPlant is here to help you solve. So what has iPlant built? Well, when we look at the cyber infrastructure that iPlant has created, there's really a variety of products, if you will. On the sort of surface layer, what we're going to cover today, what you're going to see, what most users come to iPlant to use are things like our cloud environment, Atmosphere, our web interface to the bioinformatics tools, the discovery environment. If you're using this in an educational system, in the classroom, DNA Subway, looking at image data, the BISC platform, and there are some others, but really there are a lot of forward-facing products that the average user will come to iPlant and interact with on a daily basis. However, underneath that sort of surface structure, there's a deeper structure of what we would call a middleware stack, but different technologies that most biologists probably don't want to have much to do with on a day-to-day basis. They just want to have them work. And so we have invested in making sure that these systems work together in a way that you as a biologist can pose your question, work with your data set, and then access the high-performance computing that you need, access the data sets, access the storage, without you having to do so much on the technical end of that. Practically speaking, as an iPlant user, what you get access to are platforms, tools, and data sets, storage and compute, and then also training and support, which is an example, well, this webinar is an example of that today, something that we really care about delivering well to all of our users. Another important question that I want to answer briefly in this sort of overview of what is iPlant as a project is what kind of problems can iPlant solve? Well, as the name implies, we started out as a project that serviced the plant community, and so as you'd expect, we have many products and many capabilities for looking at crops and model plant systems, but we have many users that work with animals, with livestock, with agronomic important microbes, insects. So really, if you want to think about what the answer to that question of what kind of problems iPlant solves, iPlant is built for data. So as a project, what we work with are your data sets, and whether they come from one organism or another, really the tools are largely species agnostic. So next slide, how was iPlant actually built? iPlant as a project was actually built knowing from the beginning, and this was during the era when people realized that big data in science and certainly in biology, other sciences may have been closer to that earlier on, but certainly in biological sciences we understood that big data was going to be a part of many different types of biological investigations. Perhaps the Human Genome Project was one of the launching pads for this understanding that data analysis at that scale would become routine for biologists. And we knew right away that it was not going to be a project that we could just put our heads together and come up with an answer. So we definitely needed, I guess I like this term, community cleverness. We spent more than a year and a half of intensive community requirements gathering, really asking members of the science community what products they felt they would need. And to a certain extent that's ongoing because we're always looking for input from the community. As you might expect, that resulted in us understanding what were the key workflows, pipelines, tools, capabilities that the community wanted. And as you also would infer, genomic data is the big driver behind the big data analyses in biology. So being able to do reference or de novo assemblies, RNA-seq, variant calling, ChIP-seq, those types of technologies, if you come to iPlant, you will definitely find the capabilities and resources for doing those. But one key differentiator, I think, of iPlant is that we have not just been solely about genomic data sets. As we alluded to earlier in that definition I showed, We're interested in genomic data, environmental data, phenotype data, because you really need all of those to do higher level sort of predictive and synthetic biology where you are solving those large challenges of size and scope. So whether you're looking at pathways, association, different types of modeling and evolution and ecology, you need all of those types of resources available to you. Where are we at? Because we certainly haven't finished as a project. Well, in a large sense, we think we have quite a platform for doing the genotype data. We also have platforms in place for phenotype and environmental data sets and more that are in progress. So really, we are working in many different areas along many different avenues to explore the landscape of what the community feels is important. And you as a community member and an iPlant user can always contribute your feedback as to what you'd like to see us do next. Of course, on our website, you can also get the latest update as to where we are at with various projects. I'm almost at the end of this sort of historical perspective of where iPlant came from and where we're going, but I also need to acknowledge just a few more things. One is who do we collaborate with? So it's really important to realize that iPlant as a project is not there to solve every single problem that a biologist may have. There are many other wonderful projects out there, including many of these listed who are our collaborators, where we work with those projects. In some ways we support them. In some ways they may have tools or data sets available that we as iPlant don't have. And so we want to make it as easy as possible for users to share those data. Now I see that Effia has raised a hand. If you do have a question, please type any question that you might have into the question and answer box, and I'll be more than happy to answer any questions at any time. Okay, so the next thing I wanted to comment on is who are we made up out of? iPlant as an organization is entirely virtual, so we don't have one building or one place where we just reside. iPlant is made up of 4 different institutions, with the majority of our faculty at University of Arizona, and also faculty and some of the hardware at Texas Advanced Computing Center, here at Cold Spring Harbor Laboratory as well, where we help to drive the science deliverables as well as the education, outreach, and training, and also UNC Wilmington. The other key thing for all of our users is that we're funded entirely by NSF. So this is more than $100 million of NSF investment, which means that you as a researcher would do well, I think, to take as much of it that you can use and apply it, get the use out of it, because it's been funded, it's been invested in, and really these are resources that are available for you to use right now at no direct cost. Part of our renewal was certainly a scientific advisory board that helps us keep in tune with community perspectives and priorities, as well as a focus on genotype to phenotype science. There was also additional emphasis for us on working with communities beyond plants. So as I alluded to earlier, iPlant works with multiple communities, not just with plants. Okay, so that is the history portion, if you will, a little bit about where the project came from. And now I just want to spend 1 or 2 minutes on just an overview of what are the different products that you get when you have the iPlant account, and then we'll actually do some hands-on demos. So on this slide, I've actually outlined 3 key products, or really I think of them as 3 key benefits to working with iPlant and working with the platform. Number 1, at the end of the day, we want to work with a platform that's going to get our science done. So when you work with iPlant, you have the ability to access and manage data. You have software also available that's going to help you analyze those data and the computing resources you may need to run those software and analyze those large data set. Additionally, you do have skills and help to use those software and interpret the results. This next one is very key. It's sometimes hard to notice until it's, there's a problem actually. But in many, many ways, iPlant has this baked in, is reproducibility. Working in a platform where you can manage the metadata attached to your project, be able to use open source tools. We're never interested in locking any of our users into something that they may not be able to use 5 years from now. So by using open source tools, having really broad capability to share data and workflows and metadata, it really it helps you design experiments and data management plans that are really invested in being and producing reproducible science. Finally, productivity. Being able to work with high performance and scalable computing platforms means that problems that may have taken hours, days, and weeks might take, minutes, hours, days, orders of magnitude, depending on what the original problem is. Sometimes several orders of magnitude, hundreds of orders of magnitude I've seen. So working in a scalable computing environment, which not all of us have access to, is really going to enable you to be much more productive, to automate things and spend more of your science, or more of your funding on science and not on hardware. Okay, so what do you actually get with your account? And I think this will answer the question that I've gotten, which is, do we have software or a way to analyze image data? Well, keep in mind that as a general rule, since we're a cyber infrastructure, if we use that analogy I've alluded to earlier, we could also think about ourselves as a sort of Lego building blocks or building bricks. We have built many of the pieces, and it's really up to you to decide how to put them together. So in some cases, we have built 85–95% of what you want, and almost in all situations, there are some custom things where you have particular things you want to customize, and then you build out that last 5%, or you even ask us for help to get to that last stretch. So here are some of the pieces that are key to iPlant. One is the iPlant Datastore. So this is the resource that you need to share and manage data with your lab, with your colleagues in the community. Every user has an automatic 100 gigabyte allocation, although if you want bigger allocations, that is entirely possible with a little bit of a justification statement. The data are automatically backed up, and we've made it really easy, I think, to upload, download, and share those data sets, and we'll see that in a demo in a few minutes. The discovery environment is access to hundreds of bioinformatics applications over an easy-to-use web interface. So this is a platform, and you can run almost any bioinformatics application that you would have run on the command line, except it's now in this nice web interface. So it's seamlessly integrated with our data and high-performance computing, and it's also user extensible. You can add your own application. So if you want to use a tool and it's not there very often, you can add it, or we can assist you in getting that in. We'll talk later on and do a demo on Atmosphere, which is our cloud computing resource. You have one-click access to hundreds of virtual machine images. You can fully customize those, and again, it's integrated with our other resources. If you are on the programming and systems end of the spectrum, Agave and the other science APIs really deliver a science-as-a-service platform. So you can define your compute, your storage resources, and really build your own app store, so to speak, of scientific codes and workflows. DNA Subway is wonderful for the classroom. I call it classroom-friendly bioinformatics, where we have simplified educational workflows for analyzing genomes, doing DNA barcoding, RNA-seq, and it's actually a basis for inquiry-based experiments. So for folks who are looking to do bioinformatics and wet lab activities with their classrooms, it's really an excellent way to learn. To answer the question directly was also asked, BISC is our image analysis management and metadata platform. So if you are working on your image data, first you need a place to store those images securely, analyze them. Perhaps you have your own automated software and analysis routines or APIs. All of that's possible. The software or the system natively supports hundreds of image formats and allows you really to design custom analyses. So that's a quick overview of many of the actual products that the average user would encounter when they're using their iPlant account. It's really a lot. I know that we only have an hour today, so we can only do demos on a few of them. But of course, if you want additional help, towards the end we'll also highlight how to get help, where to look for the resources, the documentation, tutorials, and that will help get you started. So for now, what we're going to do is actually do some demos. And the very first demo is going to be about how do we actually get our data into iPlant. So that's the first thing. If your data aren't there, then you probably can't go to step 2. So let's get started with that. Okay, so just a slide or two. First of all, the iPlant Datastore allows you to store any sort of data, or any type of file that's related to your research. You can move those files seamlessly between different iPlant platforms. So you may be in the discovery environment for one type of analysis. You may do something in Atmosphere for another, and you can move your data sets across those different platforms using the Datastore. There's features for automation, and again, it allows you to do sharing really nicely. Now, there are a number of ways to access the Datastore. I'm going to cover the two most popular ones. but you should know that they exist. We're going to talk today about Cyberduck, which is a little standalone download that will allow you to link up with the iPlant Datastore and move your data back and forth. And there's some other platforms, and we'll go into the discovery environment mostly for sharing. There's also the command line interface, and iCommands is the way to move data in that particular environment, and it's really, really smooth to go ahead and automate data transfers and to move large amounts of data across the networks. If you're working in the discovery environment for managing data, really that's best. You can do it with simple upload and download for small files. You can work with some things, but the discovery, you can also import from URLs, and people also ask for that as well. But the discovery environment has some size and speed limitations because you're working over regular HTTP. So this is for mostly sharing the data, I would say. Cyberduck, which we're going to use in a few moments, allows you to really almost use the data store as a Dropbox, where you can just drag and drop folders from your local computers to your iPlant storage. iDrop Desktop is a tool that we previously used and still available, but we think that the Cyberduck replaces that. So if you want to, this is available as a graphical user interface as well. I believe also for Linux, which Cyberduck is not, so some folks may be interested. And then iCommands, that interface gives you full flexibility to automate, script, and to access. Okay, so let's give you a quick demo using Cyberduck and also iCommands. So this is the point of the video where if you want to, since I'm going to be doing things on my computer, if you want to, you can go ahead and make that video full screen by pressing those 4 arrows at the top of the video window or the video pod that you're viewing right now. And that will allow you to see my computer full screen. All right, so let's talk about getting the data in. First, let me actually show it to you down on the corner of the iPlant website, here where it says Get Started under the Learning Center here. If you click here, if you forget everything in this talk, just remember to come back here because it really captures all of that. You can come back here, and we're going to focus on the second chapter of this sort of little quick guide called Importing the Data. And in order to use Cyberduck, you'll come to here and you'll just click on the appropriate download for Windows and Mac, and that's the first thing you need to download. There's actually a second thing. Once you download Cyberduck for either Windows or Mac, you're next going to have to download the Cyberduck profile, and this is just a little file that contains important details on linking Cyberduck to the iPlant Datastore. I already have Cyberduck started, or this is what it looks like once you've downloaded it and you've configured it. I'm going to go ahead and double click on this, and it's going to start to log into the iPlant Datastore and connect. That usually takes, in my experience, somewhere between 30 and 60 seconds. So while I'm doing that, let me go back to the documentation and actually show you that here in this Cyberduck link, there is something called documentation. And it will take you through step by step on the iPlant wiki all the steps you need to get Cyberduck installed. It really takes, I think, 2 minutes or so just to go through this, open Cyberduck, load the profile, and just configure it properly. So there's a lot of detail here, but it really is quite quick in order for you to go ahead and get that running. Okay, so going back to Cyberduck, that's already starting to connect. It's mounting the data store, and what's going to happen once it is finished connecting is in this window, instead of the profile and the link, I'm actually going to see the contents of my iPlant Datastore displayed with various icons. So here's different folders. I could access any of those folders and sort of see what's inside. If there's anything inside, it takes a moment. Some of these are empty. Some of these have some files in them. But it will load, and you can go through and see what folders or files there are. If there is a folder and you want to move it from your iPlant Datastore down to your local computer, as you can see, you simply drag it over to your desktop or to wherever you'd like. And as you look on the transfer dialog that just popped up, it will start the transfer in a few moments, and the download will start. Uploading is just as easy. So if I find a file on my local computer and I want to upload that, don't know if this is already in my data store, but I can just drag this over, and also that will start in a few moments. Okay, but really, that's all that there is. Once you have Cyberduck running, it's just a matter of transferring data sets by dragging and dropping them. What we've experienced is that in a good connection, you can expect to move about 100 gigs of data in about 30 minutes, which is really quite quickly. So as long as you have it configured properly via the documentation, it's quite easy to do a transfer with Cyberduck. So I don't know how to make it any more easier than that. Simply download Cyberduck and then drag and drop to move files back and forth. So all of your genome sequences, files, presentations, whatever you do that is connected with your research, you can go ahead and move those files. Okay, let me show you the command line interface. So back here, as we were just a moment ago, back in the Learning Center, there's also some other downloads. And one of the downloads there is iCommands. So you would download iCommands, and you could also follow the documentation here. I think it takes you to the tour of the Datastore, and then there's the iCommands manual and download. But follow that documentation, and it will take you through setting up and configuring iCommands. It just takes a few moments to download the software and to install it. And then there are a whole set of commands, which if you're familiar with the Linux world, those commands look very, very similar to Linux commands. So to show you how they work, let's take a quick look. I'm going to go ahead and open a terminal on my computer. And let me actually open one that has a little bit more contrast. Okay. And then if I want to connect to the iPlant Datastore, since this is all already configured, all I have to do is go ahead and say ILS, and then I'll get a complete listing of my files. Now, to answer another question I see coming across, the question is, how big can the files be that we can upload or download with Cyberduck? As far as I know, Tanya, there's no limit to the size of the files that you can upload or download with Cyberduck. I haven't tested that, but I do know that on our system, I've seen files that were almost, single files that were 700 gigabyte files. So with Cyberduck, as far as I know, there's no limitation. Of course, bigger files will take longer, but there's no limitation. If you are uploading really, really big files, it's also in many ways useful to be familiar with the iCommands because you have some more control over that. But try it, and if you get into trouble, I'll talk about how you can get help from us by going to our iPlant user forum. It's right on the homepage, ask.iPlant. But from here, you can see my iPlant Datastore. If I wanted to grab a file, I could just do it with a command called iget. And I think there's a folder. Well, first of all, I'm going to download a folder, so I need to be recursive. And the name of the folder was .vcfs. And I could download that right now. And it will start connecting to the iPlant Datastore. And looks like I already have it on my computer, so I need to force it. And in a few moments, it will start downloading those files and giving me progress, the output. This is a really, really small folder. Even though I'm on wireless, it won't take all that long. But you get the idea that it's really, really simple with a command like iget to download some folders or files. The alternative, or the reciprocal command iput, would allow you to put files from your local computer onto the iPlant Datastore. Okay, so that's a quick demo of just uploading and downloading for large transfers. What I want to do next is actually talk about how to share that data, because that's really important, sharing and also searching. So I'm going to go back to the iPlant Collaborative homepage. And again, great questions, so please, if you have any, just go ahead and keep typing them to me. Okay, so back from the iPlant Collaborative homepage, you can see on the homepage there's a link to the Discovery environment. You can do sharing of data sets in different ways, but we're going to just talk about the Discovery environment. And so from here, there's a link that's called Launch. And when you click Launch, you will be entered into the Discovery environment. I've already logged into the Discovery environment, so I did not have to enter my password. You very likely will have to enter your iPlant username and password. Okay, so this is the Discovery environment. I'm going to talk more about this in a few moments. But before I dwell on any of the other features, let me just talk about the data window. And when I click on the data window, I get a listing of all the files that I have with iPlant. If I click on my username, These are the same files that I saw at the command line, that I saw in Cyberduck, everything. Okay. I could click on a file or click on that select file, and then on the right hand side I get some ideas of when it was modified, what the size of the file is, if I'm sharing it with anybody, etc., the checksum. So all good information. Even it knows that this is a FASTA file. Now if I wanted to share an individual file with anybody, I could go up to share and then say create a public link. What this does, and when I go ahead and click on it and say create, it creates a URL that anybody could use. It doesn't have to be an iPlant user. I just go ahead and say copy that link, Control C or Command C, and then somebody clicking on that link and hitting enter, as I'm about to do there, will get the little dialog to go ahead and start downloading that file. Okay, so that's really a convenient way to share files with anybody. The limitations here are, there's two key ones. Number one, this link is not private. What I mean by that is whoever has that, and whoever they share it with, and whoever those people share it with, has a link. So if you have any sensitive data, this is probably not the best way to share that data. So if you want to revoke permission for that sharing or destroy the link, you just come back to this and you click the little delete icon in order to delete that link. Now if they've already downloaded it, they've downloaded it, but once you delete it, no one else would be able to use that URL again to download that file. The other limitation for sharing with individual or sharing files is that using this link that I just showed you in share public link, it only works with individual files. So I can't do a public link to a folder. I could compress a folder and send it that way, but that's not the way to share. So let me show you how to share any type of file, even directories or folders. I can select the folder that I'm interested in. And then once I select it, I can go up to the share and now say share with collaborators. Okay, and then I type the name or username of the person, and as you can guess, they have to have an iPlant account. It's not that hard to get, so especially if you have a lot of data, go ahead and have them sign up and then type in their name. And in a few seconds, they should come up along with their institution, and you go ahead and select that. And now this person would have read permission. Now this is described in the documentation, but I'll take you through it quickly. If I let Cornell here have read permission, then he can see that file. He can even use it to analyze and use some different applications on that file, but he can't write to it or change it or move it. If I give write permission, that means he can actually edit the file and can also deal with some of the other properties of that file. And then if I give own permission, then they have everything to do that you could do with that file, including delete it or move it. So you need to be careful. If I press done, which I'm not going to do right now, Cornell will get a notification when he logged in that something has been shared with him, and he could click on that and be navigated to it. You would also get a notification that it's been shared, and it usually just takes a few seconds. If I no longer wanted this person to have the permission, I could either come back here and change what permission they have, or I could simply click the X, and that revokes permission, and they would no longer be able to see that file. Of course, if they've downloaded it or have copied it or whatever, then they have their copy, but after you've done this with that original file, there's no more permissions since I've revoked it. Okay? The other quick features I want to show you about the files is that I can, at the top here, search in this window here. So if I type in a name, I can search for files, folders that may have that name. So I just put in a genus, which I know I have at least some data that is connected with that. Additionally, in this, files themselves, well actually if I click on a file, files are also connected with metadata. And if I go up to edit for this file, and let's see, I may not have own permission, that's why I can't do it here. Let's see. Okay. Yeah. When you go and select an individual file, files have metadata, or you can associate metadata with those files. So if I go in and say edit metadata, this file may not have anything, but I can, just for this one-off demo, add an attribute like species, and a value, or let me even say this, Dioscorea, that's the genus. Okay, you could add metadata to this file, and that's going to help with searching and organizing your data. Now, doing this one-off is not very efficient, so there are a bunch of templates. Some of them are everything from if you want to submit a sample to the NCBI SRA, which you can do through iPlant. There are a whole bunch of templates for attaching that metadata to the file. You can also use one of the other custom templates. You can still retain your own user metadata. There are also some metadata templates for working with, I'm just going to go ahead and delete this one. There are also some metadata templates for working with SRA submissions or DOI submissions, so those are some things you can do within iPlant, although we won't have time in this webinar to go through them. Okay, but once you do that, if you go up to the search and you click this little funnel, you can actually search by the metadata attribute or values. So it's really a very, very nice search feature. Okay, so that was a really pretty comprehensive overview of moving data, sharing data, and now let's actually talk about analyzing data. Let me go quickly back to the slides, just a little bit of a idea here. Hold on one second. There we go. In the discovery environment. Now, the discovery environment, as I mentioned, is a platform that you can use to run almost any bioinformatics application. It's integrated with our high-performance computing, and it's extensible, which means you can add. Now, we've already covered the first element, which was the data window. I didn't really show it to you. Maybe I will when we go back there in a second, but you can also import data from URLs. You can drag and drop folders, many of the things that you'd expect to be able to do on a desktop. You can pretty much interact in the discovery environment as necessary there. The next two features we're going to cover are the applications under the apps menu, where there are several hundred applications. You can also build automated workflows or add or integrate new applications. Unfortunately, we're not going to build a workflow or integrate a new application today. And also analyses, and we can go there to see the status of our application, our jobs really at that point, and also retrieve some of the history and the metadata from previous jobs that we've executed. If you want to see a listing of the applications that are currently within the discovery environment, this link contains it, but we'll also, again, towards the end, talk about where you can go to see that information. Okay, so let's do a demo. In this demo, we're going to do a really simple analysis that lets us touch most of the basic parts and pieces of doing any analysis within a discovery environment. We're just going to take some unaligned DNA sequences in FASTA format and make a multiple alignment. So very simple. So we'll take a look at the data. We will go ahead and use an application called Muscle to build a multiple alignment, and then we'll monitor the progress and view the results. Okay, so let's go back to the discovery environment here. And in the data window, you may have noticed, and I mentioned there's an upload feature for uploading files. So I didn't use it, but it's there. There's other things for creating new folders. So just know it's there, play around, you'll find it. But you may have noticed there's some other folders here. And in fact, when somebody shares data with you, it ends up in the Shared With Me folder. But for this second, we're going to go ahead and click on the Community Data folder, because that's where the data set is that we're going to play around with for the purpose of the short demo. And then down in iPlant Training, I'm going to click on that one. And I should favorite this so it appears at the top from now on, but DE Walkthrough, I can click on that one. All right, and so now there are some files, and including, let's see, the one that I want to use is probably at the top, DE sample plants. So here is a set of files, and if I look at it, this is a simple text file of aligned sequence or unaligned sequences. Okay, several FASTA files. Okay, so that's what we're going to use as our input. Great. So I'm going to close that, and now I'm going to open up the applications window. And currently, let's see what it looks like. There are 379 bioinformatics applications. They're sort of categorized, but you can also use a search tool. I'm going to go ahead and search for the application called Muscle, because I know it's in there. And when it comes up, I'm going to go ahead and select the one that I've already favorited. Now, before I actually click on it, just some other features. One, if you have comments or questions about the app, you can leave those there. If the app is working really well for you, you can choose to give it some stars to rate it. This is all sort of community moderated in that sense. If you're not sure what the app does, you can click on the info button. And by viewing the app info, you can get an idea, a short summary, a link to the user manual, additional information such as what was the version of that application. So all really great metadata. Excuse me. But to actually use the app, you're going to go ahead and click on the application name, and it will open up a new window. So I'll get rid of this window behind. And this is what a command line application looks like in the discovery environment. As you can see, it's a full sort of GUI, graphical user interface. So you have the option here to enter comments, which I highly encourage. We sometimes in bioinformatics, myself included, press a lot of buttons, do a lot of commands, and then don't document. So even if it's something simple, go ahead and enter those comments or those metadata. You then have a choice of where you want the results of this, this analysis output. By default, the analysis will go to your analysis folder, but if you have created a folder and you've given permission to that folder to everyone in your lab, let's say, you might elect to have your output always go there, and you can do that by default. In fact, on the upper right-hand side, there's some notifications, and there are also some preferences, and in preferences you can go and decide where your default output folder is. Okay? But we're not going to change any of that right now. We're just going to go ahead and say select input data. I'm going to click browse, and we're going to search. Now it opens to the last place that I went, but I actually want to be in a different place. DE walkthrough, DE sample plants. I'm going to go ahead and select that. Okay, this app is pretty simple. It has one more option or field. As we saw, the sequence type is DNA. Okay, and then once we're done, we click Launch Analysis. At the top of the screen, we get a notification that the application has launched, and that's really it. Once you know how to run that simple application, all the other ones look pretty similar. I'll kind of show that to you next. If I want to see what the status of my job is, I'm getting notifications, as you can see here, and I can go up there and even read those notifications. But the best way, of course, is to go to the Analyses menu. And from here, I can see what jobs are running and even a history of different jobs. Okay? As I go through my history, I may want to see, for example, well, what parameters, what files did I use in this job? I can click on that job, go up to Analyses, and say View Parameters. And I can see everything. Every option, what values I selected, even the files are here. And if I click on any of these files, it will navigate to their current location within the data store, at least where they were at the moment that you've launched them. So it's really easy to have this really valuable experimental metadata. You can even save this as a simple text file. Now, if you want to go back and relaunch an application, let's say, remember, at the command line, this is pretty easy if you want to change parameters, but clicking buttons and setting parameters with a graphical interface can get tedious. So one solution to that is to relaunch an application. If I select that application and go up to relaunch, it will open that same application window, except this time it's populated with everything that I previously used, what genome I selected, what reference annotations, the options that I had selected. Now, let's say I wanted to change this from 2 to 1. Rather than having set maybe 10 different other things, I can click Launch Analysis and redo that. But I'm not going to do that right now. I'm going to go back to Analyses, up to the top. Okay. Actually, I'm going to go ahead and refresh because that window was open and I don't know where. There we go. There we go. Okay, there's my muscle analysis completed. Once my job is completed, I can go ahead and click on the name of that job, and it will go ahead and open the results, the data. And there's the results. In this case, if I click on the, let's say, the interleaved file, there are my aligned sequences. Okay, so pretty simple. Another key thing just to remember, Log files are produced for every job, so if you have errors or issues, or if you just want to see what commands were used, all of those are in the log files, but many times you won't have to worry about them. I do want you to know that they're there. Okay, so that was a quick introduction to the iPlant discovery environment. As you saw, it's a pretty simple, easy to use interface that's entirely web-based. So you can use these bioinformatics applications without any command line knowledge, and many of us as biologists don't come to the table with that knowledge. But the next scenario, the next tool I'm going to show you is actually working on the cloud, in which case you can use graphical interfaces, you can use command line software, and we'll take you to that next step. So let me again go back to the slides and then get you oriented to the last demo that we're going to do, which is Atmosphere. Okay. And back to the slide here. Okay, so Atmosphere. As I alluded to before, Atmosphere is one-click access to these virtual machine images. You really have flexibility because you can customize the software that you set up on them, and it's quite powerful because, again, it's operating within the context of our computing and data resources. But first, let me define a couple of different vocabulary words or some technical jargon. What is cloud computing? Well, cloud computing most of us use on a daily basis. If you bank, if you go on Amazon, many things are quote-unquote in the cloud. But what does that mean? Well, let's talk about the first piece of jargon, the concept of an image, because this is how we actually store software in the cloud as a virtual machine image. Let's go, again, very simple with an analogy. You probably have had a computer and got old and you bought a new one, and now you want to take all the files from the old computer and put them onto the new system. You could take all those files off, your documents, your movies, images, data sets, and you could put them onto a backup hard drive, one by one, okay? But the alternative is actually you can make something called an image. And an image is also a file, but the difference is that an image is a complete backup of the entire system, including the operating system. Now, as you know, if you go ahead and take those documents and files and put them on another computer, you can do that with documents and files, but if you don't have Microsoft Excel installed, if you don't have Windows installed or OS X installed, you have to reinstall those software. With an image file, if you take an image, you can actually completely clone the original system. Everything down to what screensaver you use would be exactly the same. So this is the concept image, and we're going to use that concept in just a few moments. The next concept or piece of jargon is an instance. So what is an instance? Well, on the iPlant cloud, we have hard drives, CPUs, memory, RAM, all that sort of stuff. And when you come to us, you're going to ask us for a certain amount of that in order to do your science. So we would go ahead and click on how many you want, how many CPUs, how much RAM, and then you would also select an image, and that image would be what operating system do you need, what software should be installed on that, whether there's an entire pipeline and workflow there, or you want something that's clean and just the operating system. But once you put those two things together, they make what we call an instance, which is a virtual machine, a virtual computer. So in other words, when you're coming to Atmosphere, you're essentially creating a computer that is a remote one, but you're creating it on demand, which means when you need it, you can use it, and when you don't need it, you return those resources to us. When you're using Atmosphere, you can choose an existing image that may have software installed. You may customize that image. You can have up to 16-core, 128 gig machines, which off the shelf would be pretty expensive if you were trying to do that and just purchase it from Best Buy or something like that. As you're going to see in a moment, we can use that on the shell or via VNC, so we can see a desktop. And here's a really important thing, I think. You can also share your image with other users or even make them public. Okay, so what are some use cases for Atmosphere? Well, one, you can run software and analyze data sets that currently you only run on a laptop or a desktop. So if you're running something and it takes several hours, why occupy your computer locally when you can use an Atmosphere instance? You can also run visually oriented programs. Those are graphical user interface. In the discovery environment, if you had a piece of software that had its own graphics, you would need to use, you couldn't use a discovery environment, but you could use the atmosphere. You also have something called pseudo access, which means you can manage complex dependencies. You have all the administrative rights. You don't need someone else to install things for you. You also can use atmosphere, and this is one of my favorite uses, for teaching and for working in the lab with collaborators or students, because every instance is the same. So everyone will be working with the same software installed, the same data sets available to them. And if you're developing software, scripts, pipelines, tools, you can also release them as an instance or as an image, and everyone can go ahead and use them. Okay, so let's take you through a quick demo. If you want to work on this demo, if you want to connect, there are some software requirements. On Windows, if we're using an instance that has a desktop, you might want to download VNC, and that's actually true for all the platforms or any VNC viewer. The SSH, if you want to actually log into your instance on Windows, you might use something like PuTTY, and Mac and Linux, you can use the terminal. Okay, so let's give you a quick demo. Let me go back to the iPlant Collaborative homepage. And from here, we can go ahead and click Launch to get into Atmosphere. Okay. Now I already have Atmosphere open, and this is something what it looks like. Except when you come, you'll be asked to launch a new instance. So I'm going to go ahead and click Launch New Instance. Now let me also remind you, access to Atmosphere is not automatic. So let me just go back to the home page for one moment. When you log in and you go to the user portal, as you signed in the very first time you got your account, you need to request access to Atmosphere. So in the user portal, I have Atmosphere as one of my services. If you're starting with iPlant for the first time, it'll likely be at the bottom under Available Services, and there'll be a link there to go ahead and request access. You must have an educational, a .edu or a .org account. I think also .gov accounts if you're working in something like USDA in order to get access. So not your personal Gmail, basically. Okay, so back to Atmosphere. In Atmosphere, I have a listing of these instances. These are complete software setups. There are operating systems, there are data sets perhaps, there are software, and you can see everything from somebody who has an astronomy image analysis, okay, great, to if I search for something like RNA-seq, I can start getting different things from Trinity to Ubuntu RNA-seq with whatever types of tools installed. Once you find one that you like, often there's a description there as to what's on there and maybe even some help. And then you select what amount of resources you need. Now, an important caveat, well, two important caveats. One, the biggest systems, and this, it's not always available at every single moment, so it depends on the capacity of what's available. Additionally, you have an allocation. So as you're using resources within iPlant, if you exceed your allocation, you'll either need to wait till it refreshes, which it does on a monthly basis, or you'll need to request more resources. But once you've selected it and you've clicked Launch, in a few minutes on the left-hand side, you're going to see an IP address which you can use to connect. Once that status is green, you'll be able to connect to it. I'm going to go ahead and open VNC, which is that download. And it already, I think, has that address in there, so all I have to do is click Connect. I enter my iPlant username and my iPlant password. And in a second, I'm connected to a completely virtual remote computer. And anything that I would do on my local desktop, I could do. There are some software installed, so you can see there's FastQC there. I could start that program and start analyzing some data with FastQC. This is a graphical program, something like RStudio. I could open the terminal, and that's all there. Now, one important caveat is that when you start an instance on Atmosphere, it does not have access to your iPlant Datastore. Your data that you saw is not there, as we saw it in Cyberduck, a discovery environment. So the very first time you start it, you need to bring data over. You can do this in a number of ways. iDrop is installed there. I could, iCommands, if you are familiar with the command line, you could start setting that up with an initiate command and go ahead and enter the fields that are in documentation. But you'll have to bring those data sets over from the iPlant Datastore. Now, in addition to connecting via VNC, you can also connect via the terminal. Let me go back and get that IP address. Okay, and if I open up a terminal, okay, I can do SSH, my iPlant username at that IP address, and I'll go ahead and say yes, enter my iPlant password, and now I'm connected. If I want to sort of, test that, I can make a folder on the desktop. It should appear there, okay? So either way, whether you're doing it through VNC or whether you're connecting to it by SSH, you can connect to your Atmosphere instance. Now, as you use your Atmosphere instance, your allocation, your number of hours will start to go. And again, it will stop once the instance will stop until once it runs out, until you refresh. You could also go to that instance and actually pause the instance or stop or suspend. If you have an issue, you can reboot the instance. If you have installed some software, you think it looks great, you can image that instance, and then now the next time you come and use it, that software will be installed. You won't have to reinstall it. It'll all be there. And when you create an image, you can decide whether that's private to you, available to a select group of people, or entirely public. Once you're done, finally, you can go ahead and terminate that instance. Termination is final. If you have data on that instance and you haven't copied it back to iPlant or downloaded it to somewhere else, everything will be deleted. So don't click terminate until you're really sure you are ready to terminate. So I'm going to go ahead and say yes, because I'm not using this anymore. And my instance will go ahead and terminate. Okay, so that's all that I really wanted to show you for today. The very last thing that I'm going to go ahead and show you is where to go for help. And right now, if you have any questions, go ahead and start typing those. If we get to the end, we don't have any questions, I'll assume that you got what you needed, at least for right now. The entire presentation is already being recorded, so usually within a day or so, this will be on YouTube and you'll get a notification when it's posted. So you can go back and review or even share this with any of your colleagues. Back on the iPlant homepage, let me show you this really quickly. In fact, there's some slides for it. Where to go next. Okay, so in terms of where you want to go next with this, on the iPlant homepage, on the bottom right-hand corner, there's actually the iPlant Learning Center. And there's also a list of upcoming events, including workshops, webinars. You can request workshops and webinars from us. I want to also give an acknowledgement to our executive team and many, but not all of the people who worked or work at iPlant who contribute to what we're able to do today. And again, just to emphasize and show you that on the iPlant homepage, another important place to go is in the Learning Center, Ask iPlant, and this is a user forum. Post any question here. In fact, as you're posting, you may find out that that question has already been answered. So go ahead and use that as a feature to get help. Thanks very much for joining us, and thank you for signing up for an account.